Hit record. We're going to take just a couple seconds of silence, and then we'll jump into it. Optimal minimal.
At this altitude, I can run flat out for a half mile before my hands start shaking.
Can I answer your personal question?
Now would seem an appropriate time.
What if I did the opposite?
I'm a cybernetic organism, living tissue over metal endoskeleton.
The Tim Ferriss Show.
This episode is brought to you by AG1, the daily foundational nutritional supplement that supports whole body health. I do get asked a lot what I would take if I could only take one supplement. And the true answer is invariably AG1. It simply covers a ton of bases. I usually drink it in the mornings and frequently take their travel packs with me on the road. So what is AG1? AG1 is a science-driven formulation of vitamins, probiotics, and whole food sourced nutrients. In a single scoop, AG1 gives you support for the brain, gut, and immune system. So take ownership of your health and try AG1 today. You will get a free 1-year supply of vitamin D and 5 free AG1 travel packs with your first subscription purchase. So learn more, check it out. Go to drinkag1.com/Tim. That's drinkag1, the number 1, drinkag1.com/Tim. Last time, drinkag1.com/Tim. Check it out. As always, you can subscribe to this podcast on iTunes, and you can find all of the links and resources from this episode, as well as every other episode, by going to 4hourworkweek.com/podcast. Spell it all out, or you can go to 4hourworkweek.com and just click on podcast. Feedback, if you have feedback, I would love your thoughts, anything at all, who you'd like to see on this show. Ping me on Twitter at tferriss, that's twitter.com/TFERRISS, or on Facebook at facebook.com/Tim Ferriss with 2 R's and 2 S's. Hello, my clever little monkeys. This is Tim Ferriss, and welcome to The Tim Ferriss Show, where I dissect world-class performers, interview them, not literally cut them apart and dissect them, to try to extract the tools and tips and to find the resources that you can apply in your daily lives, whether those people be billionaire investors, chess prodigies, famous CEOs, celebrity types, or anybody in between. It's really a wide spectrum of expertise, and you find commonalities. And in this episode, we will be talking to 2 scientists/entrepreneurs, specifically about the microbiome. And many of you, literally hundreds of you, have asked me to elaborate on the microbiome and what that means and how I try to manage or improve my own microbiome. And we have 2 people on this particular episode. We have Jessica Richman, who's co-founder and CEO of uBiome, uBiome.com, a startup backed by Combinator and Andreesen Horowitz, which uses citizen science to understand the human microbiome. In full disclosure, I am also involved with this company. I helped them long ago and then ended up only recently backing the company. So we will talk a lot about that. And then you have Jonathan Eisen, who is a full professor at the University of California, Davis, with appointments in the School of Medicine and the College of Biological Sciences. Dr. Eisen's research focuses on the evolution, ecology, and function of community of microorganisms. Both of these people are fascinating, very different personality types, and the conversation was a blast. So without further ado, please meet Jessica and Jonathan. Jessica and Jonathan, thank you so much for taking the time to be on the show.
Glad to be here.
Really honored to be here.
And this is a distributed powwow. I'm very excited about this. My first time doing this. Jessica, you are here in San Francisco?
Yes. Yeah, in San Francisco.
Right down the street. And Jonathan, you are in Vegas. So
I am in Vegas. That's right.
In Vegas to settle some old debts, to settle some old scores? No. What are you doing?
Yeah, I'm doing a show. No, I'm going out to do field work in Death Valley tomorrow.
All right. So this leads us to the very exciting topic, and many topics I'm sure that we'll delve into. I will start with a very boring question. It is perhaps the most American of all questions, but the what do you do question. But very specifically, I'll start with Jessica. If somebody asks you these days, I guess your answer is it might be somewhat straightforward, but what do you do? How do you answer that at a cocktail party, if they still have such things?
So, I mean, I don't know if I've ever actually been to a cocktail party, but when I go to conferences, people ask me that question, and I have to back up a bit and start talking about bacteria. And then either people get this really horrified look on their face, or they get this very excited look on their face. Most people are not neutral about bacteria. So I say, oh, well, I run a company that where we sequence the microbiome. Those are the trillions of bacteria that live on and in our bodies. And then I sort of pause and wait for the look of horror or excitement to cross over their face to see how much more I should tell them about that. And that's a matter of time before we get into the subject of poop.
I knew you were gonna bring it up first. This is a very
there's like a mean time to poop for a conversation.
Yeah,
basically.
you're like, so how much do you know about poop swaps? That's the opener. I find that's a pretty good pickup line, also.
Yeah, it works for me.
So, Jonathan, what about yourself? When someone asks you what you do, what is your answer?
I'm a professor, and I study and teach about and communicate about microorganisms.
And I want to define a couple of terms, partially because in all honesty, and this is embarrassing, but I've never really defined these terms very well for myself. Microbiology. So you're currently a full professor at UC Davis, is that right? At the UC Davis Genome Center. What is medical microbiology and what is microbiology? What's a good way to think about that?
Well, microbiology is the study of organisms that we can't see without the aid of like a microscope, some device. They're organisms that are invisible to the naked eye. Mm-hmm. And then, within that scope of microbiology, there's lots of sub-disciplines that people have defined. I kind of hate all of those sub-disciplines, but it's, some people, environmental microbiology is the study of microorganisms out in the environment, and medical microbiology is the study of microorganisms associated with human health, and there's veterinary microbiology and plant microbiology and blah, 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 blah.
Now, do you dislike those classifications because it's kind of like saying breast cancer versus pancreatic cancer versus such and such cancer, and you're like, we should really be defining this by the type of microorganism, not the location, so to speak, or is it something else that bothers you about it?
I mean, in general, I hate rules and I hate stovepiping, so I just think it's not that useful to isolate humans from other animals and to say that environmental microbiology is somehow different than the microbiology inside people. It's just arbitrary boundaries that have no value.
Okay, I like this. This is a good start. And we will certainly talk about it. You have a very interesting blog. What is phylogenomics or phylogenomics? I can never figure out.
Yeah, phylogenomics.
Genomics. There we go.
It's both a good and a bad thing. I invented the word when I was a graduate student to basically refer to evolution. Philo is a sort of abbreviation for evolution, and genomics, the evolution of genomes, basically. And I regret this because I also write in my blog a lot about the proliferation of bad new genomics words. And but nevertheless, I invented the word, and then I just named everything, all my blogs, all my lab sites, all my login accounts to, various sites. They all have something to do with phylogenomics, and I'm kind of stuck with it.
Well, you own it. That's good. I mean,
that is true. I own it and don't like it.
and the evolution, so the microbiome, and I'm wading very deep into my into the pool of ignorance on my part when I get into the microbiome, but it became of great personal interest to me because I contracted Lyme disease, went on very long-term, from my perspective, long-term use of broad-spectrum antibiotics like doxycycline, and started to suffer from all sorts of health conditions that from my assessment, were not caused by Lyme disease, but were caused by the long-term use of antibiotics. So I started to have skin disruptions and all sorts of issues that are associated with chronic illness. And but I had not drawn the connection between some of these supposed chronic illnesses and microbiome. A friend of mine, who is sort of an amateur microbiologist, for lack of a better term, suggested that I start taking L-plantarum and a number of other types of probiotics, and the symptoms, I had this breakout on my feet, pretty much overnight down by 50% in terms of symptoms, and then proceeded from there really quickly. And so I'd love to know, what are the biggest misconceptions? And this is open, maybe Jonathan, if you want to take a stab at this, what are the biggest misconceptions about the microbiome? Because it's become a hot topic. Obviously the fecal matter transplants are very exciting for the media. And, that would lead me to assume that there's a lot of voodoo and nonsense also being spread, therefore. But what are the biggest misconceptions about the microbiome?
Well, I mean, I think that the thing that the reason I am interested in this from a research and a communication point of view is because there's enormous promise from medical and agricultural and environmental and evolutionary point of view. I mean, every plant and animal is covered in a cloud of microbes, and they clearly influence the biology of these things that they're living on. And so there's enormous promise, and there have been a few papers in the last 5 to 10 years that have shown really big impacts of this cloud of microbes on the health of various organisms. But the part that is also interesting and sort of challenging is, for whatever reason, a lot of people have oversold or overinterpreted what we know about the microbiome and aren't doing a really good job of distinguishing what we think might be going on from what we know is going on. So I give out an Overselling the Microbiome award on my blog, and it's like it's literally like, the easiest low-hanging fruit blog material that exists in my life. There's just so much out there where people make sort of fundamental miscalculations as to how to interpret some scientific study or some personal dietary change or other issues.
Well, I mean, I think I'd like to add a couple of things to that. I think the first thing is that, you've noticed anecdotally this change, Tim, right? Like you take all these antibiotics, you develop this problem, you take probiotics and it goes away. And that's not data, that's just an anecdote. But I think...
Yeah, and just in fairness, I recognize that it could be correlation, not causation. Yeah, it could be regression to the mean. I understand all that.
But still, I mean, I don't call it an anecdote to minimize it, but just to say that I think one of the interesting things that we're doing at uBiome, and that I think is a general trend that is interesting, is this crowdsourcing of science to enable things like what you experienced to be validated in new ways. Because I don't think it was my feeling about it probably was not just regression to the mean if it happened immediately afterwards, and you can sort of see the effects in your own experience. But why did that happen, and does it happen for everyone, and how often can that be replicated, and all these kinds of things are important questions to be answered. And your anecdote is like the beginning of a hypothesis about how probiotics can affect the human body, not just something to be dismissed, I think.
No, absolutely. I mean, I'd love to hear you elaborate a little bit on how you would define or think of citizen science, because I think we're leading into that pretty quickly.
Yeah. So citizen science is a term that was coined by the Cornell Ornithology Lab in the 70s. It's basically the idea that citizens, people that are not Ph.D. scientists working in a research lab, can create interesting science by collaborating with scientists. So in the case of ornithology, it's really interesting. There aren't enough bird watchers there aren't enough ornithologists in the world to watch all the birds. So they use amateur bird watchers to contribute data very frequently. And that's sort of grown in this idea that other types of scientists can include the public in their research, whether it's data collection or even going further, it's generating hypotheses, funding science like we did at uBiome, open sourcing publication of blogs about their own experience, their own experiments. And all of these things are sort of citizen scientists, crowd scientists, people who are not employed in the job of science, contributing to science and adding a new perspective that isn't there in mainstream science.
Got it. And I want to come back to Jonathan. I'm going to ask Jessica a few more questions, and then I want to come back to you and ask you. So you have so much experience with research. I'd love to ask you about sort of what is wrong with the current ways in which research is done or has to be done, and how you, in a dream world, would fix that, because I think it's an interesting topic. But Jessica, I'd love to ask you just to give people sort of a taste, and again, understanding that the plural of anecdote is not data, although I do have some issues with the overuse of that expression.
I do too.
I'd love for you to comment, because we've talked about some of these things before, obviously, and I'm involved with, I'm a backer of uBiome and believe in the mission, and I was a supporter long before I was ever involved in a formal capacity. But could you talk about gut, mood, and behavior, maybe highlighting some of the things you've found most interesting in the last few years?
Yeah, so there's a lot of so let me just back up for a second. So the obvious things that you think of when you think of the gut microbiome are, health conditions that involve the gut. So you think about, traveler's diarrhea, and you think about enteric dysfunction, and you think about irritable bowel or Crohn's disease, or all the sort of obvious things that involve your gut. But what you don't often think about are other health conditions that are much more systemic that relate to the microbiome. So there's been some interesting research about, mostly in mice, about how you can change a mouse mood by changing the mouse microbiome. And I know this sounds a little nuts, but there are measures for whether a mouse is anxious or depressed, or has autism. Actually, it was an interesting study I was just reading earlier today. And basically by taking germ-free mice, these are mice without any microbiome, and then by adding either an anxious microbiome from an anxious mouse or from a not anxious mouse, you can change the mood of a mouse. And this has been done in a number of different ways with other sort of mood disorders in mice. And there has been human research specifically on this. I mean, they notice different microbiomes in humans that have different mood conditions, but there's no causal research there. That's a really interesting area. And I think it's interesting mostly because it points to how complex the microbiome is and how complex its effects are. So it's not just, the obvious, you have diarrhea and that's because you have a microbiome problem. It's much more dispersed throughout the body, and the effects the microbiome has can be much more subtle.
Well, this comes back to something that Jonathan said about distinguishing between what we truly know and what we think we know, and there's often a very large discrepancy between the two. I mean, for a long time, it's thought that fat cells are basically these inert storage devices without much function beyond that. But a lot of endocrinologists have begun thinking of adipose tissue almost as, it is basically hormone producing endocrine glands in a way, and it's very active. I mean, there's brown fat and different types of adipose tissue as well. And I think that one of the things I've heard from some scientists I've chatted with about the microbiome is that the gut can be thought of as the second brain. And where I've lost track of the argument is how the gut, if it does in fact produce neurotransmitters like serotonin, or is it just that the composition of the microbiome in the gut then affects brain function in such a way that affects neurotransmitter production? Do you have any thoughts?
I mean, I think it's all of those, right?
Yeah. Yeah, so there are a few hypothesized mechanisms, and Jonathan, maybe you can expand this more than I can, but to start off with, there's this idea that that microbes cause inflammation in the gut, and then your brain misunderstands, feels that you're kind of out of sorts and inflamed, and then is anxious about that, and then you sort of assign that to your work situation or your romantic situation, where really you're just kind of irritated because your gut is irritated. So that's one hypothesis.
Right.
Another one is that the microbes are actually making chemicals that induce behavior in you, so they're making you crave sugar because they want sugar, not because you need sugar. So maybe even making you crave things that you can't digest, but that they can. So this is a matter of survival for the gut microbes to, the only way they're going to get fed is if you eat what they want you to eat, right? So that's sort of another idea.
They're pulling a Feed Me Seymour sort of
exactly.
So I wonder if we can take a step back for a second. So I've been, and many other people have been working on how host, animals and plants depend upon single bacteria, single mutualistic, beneficial symbionts in a variety of ways. And we know from diverse studies of plants and animals that, lots of insects need bacteria that live in their gut to make amino acids and vitamins they don't get in their diet. And lots of plants get nitrogen via bacteria that live inside their roots. And organisms that digest cellulose, like termites, the microbes are doing it. And there's just hundreds, thousands of examples where a microbe is providing some critical function that the host is not doing for itself. And there's also, of course, we know lots about the hundreds to thousands of examples of pathogenic, dangerous organisms that, cause some disease or some problem where they manipulate the host biology in all sorts of bizarre, interesting, and, damaging ways. And so in that context, it's almost obvious that the microbes that live in our gut and in our mouth and on our skin and in our other orifices and places have the potential to do all sorts of things to our biology. And again, the way I think about it, I'm actually an evolutionary biologist. The way I think about it is, animals have been evolving in this microbial world for hundreds of millions of years. Our evolutionary processes are in essence based upon expectations, and the expectation for our gut and our immune system and our behavior and everything is that we're going to encounter microbes because we always do. Mm-hmm. And so our systems are tuned towards, predicting and responding and dealing with microbes. So, in that context, it's almost obvious that microbes that are in and on us are fundamental parts of our lives.
And to give, I'd love to do a quick fact check. I've heard people say that humans are really 10% human, meaning by volume or number of cells, 90% of us is comprised of bacteria. Is that? Yeah,
there was a really good fact checking that someone finally did in I think in the Boston Globe, where it turns out that number, which had been quoted for 20 years or so, is almost completely made up. But it's more, better estimates are probably something on the order of 50% microbe. But I don't think I don't think that the total number of cells is that interesting. What I think is more interesting is that in our mouth there are probably, in each individual person, there are probably something on the order of 500 species or 1,000 different species of microbes. And in our gut, there are, 1,000 in each different compartment, and they're not always the same. So the things in the stomach are different than the things in the ileum, are different than the things in the colon, and then on our skin and in different parts of our skin. We are an ecosystem with an incredible diversity of types of organisms and functions. And each person, it's going to vary how many they have, how many cells there are. It's going to vary over time, but we should view ourselves as a walking ecosystem, not as an individual human.
And how if people wanted to learn more about the well, actually, let me take a step back. I promised I would ask a question, so I will ask that. And that is, for researching the microbiome or anything else, how is it currently done, and how would you change it? What are the issues with accelerating good research in this field or any other for that matter?
Well, I think the reason everybody is excited now, the reason that Jessica was able to start her company, and the reason that there's a lot of stuff going on, is that the technology for studying microbial communities has gotten much better and much cheaper in the last few years. And primarily the way people do this is using analysis of the DNA from a sample. And the reason we do that is most of the microbes in any particular environment can't be grown in the lab, and we can't identify them very well in a microscope. But we can crack them open, and we can look inside of them, and we can look in particular at their genetic material, their DNA. And that DNA contains a lot of information about the microbes that are there, that helps you identify what kinds there are, and it helps you predict what their functional potential is. And so in that context, it's like I'm like a kid in a candy shop. It is the golden age of microbial ecology right now. So I wouldn't say there's a ton per se I want to change. What I really want to do more than anything else is engage the broader community in thinking about microbes, just like Jessica is trying to do, we're limited by the number of samples we can get.
Right.
And what we want is 100 million samples, to be honest. And I can't do that myself. I need 1 million people to help me collect samples. That's what I want to get people to think about.
I agree with that. I mean, I would say I have a lot of thoughts on this because it's something we think about a lot, and how to make research accessible to the public, how to make it larger, more scalable, more actionable in people's lives. And I think larger studies is a huge one. I think that, you can have a very respectable study in a major peer-reviewed scientific journal, Nature, Science, Cell, something like that, and you can have 100 people in it. And I just think that that's going to seem ridiculous 10 years from now, 15 years from now, when it's just obvious that you can source study participants more easily and that you can involve the public in your study and just have things move much more quickly. So I'd love to see that happen. I'd love to see studies be more scalable and flexible so that, you can start off with a certain study design, asking a certain question. And then because you have, your 10,000 participants or your 100,000 participants, you can sort of take a subset, say that's the interesting part, and then go forth with that group. So you can sort of make the whole apparatus of creating scientific studies better, larger and more scalable.
And I was just going to say, I mean, I think that the one thing that maybe I would say we need to do in the future that we're not doing as much of now is that a lot of the work that we're doing, it's incredibly exciting and really interesting, where we can use DNA as a fingerprint of what's going on in the community. But this is building, in essence, some type of general profile of the world around us. And we can compare and contrast health and disease states. We can compare people over time. We can compare people before and after, like you, antibiotic treatment. We can do lots to get a general idea as to what's going on. But of course, that's just the first step. I mean, there's lots more really interesting biological studies that can be done once you have this sort of framework that comes from characterizing the DNA of the community. So we can start to look at the functions actually encoded in these genes. We can do experiments to test those functions. We can look at where individual cells are located in particular environments. So, a fecal sample doesn't tell us anywhere near as much as a full microscopy staining of the entire gut. And, things are floating around in the liquid. Some are on the layers of cells. Some are inside cells. I mean, this is the first step in characterizing this system.
So, I'm sorry, I was just going to ask quickly, just a couple of observations for people who are listening who are from a lay audience like I am. I'm not a trained scientist. I mean, I read just enough to be dangerous, and I've spent some time at UCSF with a couple of the neuroscience labs, but I think that I want to come back just briefly to the plural of anecdote isn't data, because this is a term that is used a lot by people who don't really understand the design of scientific studies. And I think it's worth just pointing out a few things, and please jump in and correct me or edit anything that I say. But if you look at, for instance, the development of smallpox vaccine, which was introduced by Edward Jenner in 1798. So he observed, and one could say this is anecdotal, that milkmaids who had previously caught cowpox did not later catch smallpox. And the way he demonstrated this could be causal is he, as I understand it, basically took cow pus and injected into his family. And needless to say, pretty ballsy. But the point being that the observation helped him form the hypothesis, and that could be derided by this. The plural of anecdote is not data. And when there's a study, I mean, you brought up 100 subjects, right? So oftentimes people from a lay audience will say, "Oh, there are only 10 subjects. Like that's a bullshit study." And it's like, well, there are a couple things you have to look at, right? You can look at the number of the N. You can look at the number of subjects, but you have to also look at the amplitude of the change from the intervention, right? If you're doing a control and experimental group. So if you have 10 people, but they all double their working memory, well, okay, there might be something interesting there in terms of the p-value, right? And the likelihood that that's attributable to chance. is rather low.
I'd say, Tim, like,
Yeah.
I think that's true. I would say the plural of anecdote is hypothesis more than it is data, right?
Yeah.
I mean, that's kind of the example that you're giving. But,
Well, right. No,
Well,
but,
so I mean, I think that, I mean, my personal view on this is that many scientists, many of the supposed cognoscenti, just are assholes. I mean, and let's just be clear. I mean, you know, like, they say things like that because they want to put themselves up on a pedestal. And I think that if you go back to your question about the future of experiments and science in general, I mean, I think what we need in the future is for much more of these observations all the way through to clinical trials to be published and to synthesize the collection of knowledge from across the planet more than we need suppression of work that people do simply because one person thinks it doesn't have enough statistical sample size or something like that. I mean, that is the ultimate. And in fact, I spend half of my time working on scholarly communication and open science, not on microbiology. And I think that this is the biggest issue that we have right now, is trying to broaden scientific research and not to suppress it. And it drives me crazy. It makes me just go insane when I hear anyone say anything like that quote. And I hear this all the time in, you know, graduate student committee meetings where one person will say, well, that's not hypothesis-driven research, that's, you know, discovery science. And, you know, God, just get out of science, for God's sake.
This brings up a follow-up question I'd love to ask you, which is if you had, let's just say, $5 million to use as you see fit in science, whether it's for the sort of microbiome specific stuff or the open science initiatives or both, how would you spend that? If you just had carte blanche, here's $5 million, assuming it's not to run off to Monaco and, you know, buy Lamborghinis, you can use this for almost anything you want. How would you spend that? I
would invest it in lots of small projects.
What types of small projects?
You know, I think that what I see is that creativity by researchers, by citizens, by graduate students, by undergrads, by high school students, the creativity that people have about science, about microbiology, once they learn about microbiology, is enormous. And what we need to sort of support is ways that those people can leverage their creativity to do a research project. And what I would not do with the money is say, give one person the $5 million to do a big project. I don't think we have a lot of those projects anyway. And what I really think we need is to harness sort of the anecdotes, the observations that people have made. They may not all turn out to be useful interpretations, but there's a lot of stuff out there that needs to be studied, and we need more people doing it.
Yeah, I think it's interesting. If I had $5 million that someone handed me to do this kind of research, I think you're right. I would break it down into small chunks. I think that makes a lot of sense. You sort of let 1,000 flowers bloom, sort of see what comes out. But I think in terms of the microbiome, what's most interesting is to accelerate the process of taking these sort of correlational studies that we have now, which say, oh, isn't this interesting? People with X health condition, whether that's anything from Crohn's disease to autism, people with that health condition are different than people that don't have that health condition. I think that's sort of the state of a lot of the research, and I would use that money to do a number of studies that would move the research to the next phase, which is great. Okay, where's our diagnostic and where's our therapeutic based on that? Because I think the real power of the microbiome and what's sort of magical about it is that these microorganisms are both sort of biosensors that can tell us what's going on in the microbiome. At the same time, they're also drugs, right? Like, they are organisms that when you put them in the ecosystem of our body, change the outcome from a clinical perspective. And I think that moving things from this sort of associational stage to a specific I don't know, you could call it a product stage, but to a specific improvement in outcomes. I think that's going to happen anyways, because a lot of people do have $5 million and they're spending it on that, which I think is great. But I would also take my $5 million, and knowing what I know now from working on uBiome, I would put that to good use in those areas.
Well, I think the micro experiments are really undervalued because for whatever reason it seems like a lot of the scientific community undervalue the things that cost very little money just because of advances in technology. And for instance, I mean, and this might seem like a silly example, but the late Seth Roberts, very smart guy. I mean, very good at crunching numbers and spotting methodological flaws. But he introduced me to this very simple, and there was he did a fair amount of literature review to try to figure it out, but just the combination of vinegar plus honey in hot water before going to bed. And I was able in the span of about 48 hours to have several thousand readers test this. So people who had insomnia to test it and compare it, obviously subjectively, sort of qualitatively, with other things they had tried, like Lunesta or whatever. And it was astonishing how positively people responded. But I struggle with how to sort of gather that and codify it and present it in a way that can be graphed intelligently, right?
That's absolutely it. Yeah. I mean, right. Because that's a valuable study. If you just put a hypothesis at the beginning and a publication at the end, there's some, that's a scientific study. You just have to sort of structure it properly.
Exactly. And I think what's exciting to me, at least, is I have, a captive audience of 1 million plus people per month, and you can compensate for a lot of flaws in the data with large numbers, right? With the law of large numbers. If you have enough people, you can smooth out a lot of the rough edges. Let me dig into a slightly different question, and Jessica, I'll toss this to you first. A lot of people think about probiotics, right? And there's obviously a huge industry around probiotics. I think some of it, even from my untrained standpoint, look pretty nonsensical, or they're pushing the claims really far, or they're using for those people out there who don't know the term puffery. I don't know if you guys have ever heard this expression. So there's a legal definition. Puffery is when you make claims that are nonsensical. They're non-falsifiable. So if you buy a shampoo that says, beta hydroxylizing hair volumizing formula, that's puffery because it's all fucking bullshit. And that kind of stuff is used all the time in supplements, right? It's like, increase your vitality and blah, blah, blah. Okay. Vitality, puffery, bullshit. But what I wanted to chat about, because I had this planted in my head by, a friend who's, he's an amateur scientist, but in the same way that I'm not going to say Darwin, but I mean, people who ended up coming up with pretty fascinating things were amateurs. And he talked about the importance of, more so than probiotics, basically treating your, for instance, gut microbiome like a rainforest, where you need to create an environment in which plants can grow. You can't just stick plants into the sand and hope that they'll take root. And so he has focused quite a lot on foods that have, whether it's say beans, lentils, things with fiber, or prebiotics like baobab root and so on, to help the body create an environment in which beneficial bacteria can thrive, as opposed to swallowing a lot of pills with said bacteria within them. And again, all of this might be BS. I don't know, but how do you guys think about this? And Jessica, I'll let you go first.
Yeah, let me start with that one. This is a great topic. I'm really glad you brought this up, because I have a lot of thoughts about this. So the first thought is, to some degree, people have to use that language of puffery because for legal reasons. I mean, they can't say, this product is going to make you healthy in these specific ways. They have to say increase vitality, which is total nonsense.
Well, they can, but it's just expensive to get FDA.
Yeah, that's fair enough. It is expensive. Yeah. But I think people also use that puffery because I hate to say it, but the current state of the probiotics industry is that if you have a study that shows that your probiotic exists in the bottle and is taken by the people that take it, and shows up somewhere in their stool after they've taken it, that is a gold standard amazing study.
Wait, that's like if it's excreted. Meaning,
Right.
like the Olympic athletes having the most expensive urine in the world.
Right,
kind of thing.
exactly.
Yeah, okay.
It's really, I mean, I think this is, I mean, no fault to the probiotics industry here. This kind of testing to better understand the microbiome is only possible in the last few years. So this is why they haven't done this yet. But
Right.
this is a over $30 billion a year industry. It's a huge industry. And basically the science supporting it just hasn't been there. So I was at this conference, that was a Nutrition Business Journal conference, and there were all these executives there from, the nutritional supplements industry. And they're just starting to figure out, like, how are we going to, in an age where everyone can test things about themselves, the age of the quantified self, and in an age where DNA testing of bacteria is pretty much ubiquitous, and we charge $89 each. So that's like really cheap. You can do a lot of, everyone can basically test themselves. How do they make sure that their products are actually doing something and are actually valuable? And so I think there's going to be I think the probiotics that you see on the shelves today in Whole Foods are going to be totally different 10 years from now, because the public will just demand better. They'll say, well, wait, I took this and what's it doing for me? And because of science, it'll be possible to do better. So I think there'll be the probiotics industry is poised for this tremendous leap into better products that actually do a lot more for you. And then can be tested sort of apples to apples with prebiotics, like you said, like are prebiotics better? Well, it probably depends on who you are personally. It depends on your health conditions. It depends on sort of, are the probiotics that are written on the outside of the bottle actually in that bottle? That also is a big,
there's
big
that
factor.
too. Yeah.
I mean, it sounds silly, but that is actually a huge factor, right? Like, if they're not, there's currently no sort of testing for what's live. So I think there's going to be a big change in that industry as these new technologies sort of work their way through and people develop better products going forward.
Yeah. Did you buy an expensive bottle of dead sea monkeys?
Yeah.
Yeah. I saw. I'm sorry, Jonathan.
Yeah.
Go ahead.
No, I was just going to say that, I think the growing appreciation of this sort of cloud of microbes, the microbiome, that it's an ecosystem, or at least it's complex like an ecosystem, is, there are ways that we should be thinking about this ecosystem that probably, the simple model of probiotics just didn't take this into account. I mean, really, we need to think about the dynamics of competition going on in the ecosystem, of the different members competing with each other for food and for space. They're going to grow and reproduce at different rates within the system. But just like any other ecosystem, there's going to be influx, that is new organisms coming into the ecosystem. There's going to be efflux, and there's 200 years of ecological research on studying these things, and probiotics are have the potential to impact the system, because if you have a massive influx of some individual species into an ecosystem, it can impact it either on the short term or the long term, but it's a very simple component of the total picture of the ecosystem. So just a good example of this is if you take a savanna or a chunk of savanna and you isolate it, and you introduce into that savanna 1,000 cattle, they're going to disappear pretty darn fast. But if you introduce 1,000 zebras, organisms that, roughly the same type of organism, but they're kind of adapted to living in that ecosystem, they're going to do differently. And if you introduce, 50 species at a time that actually represent something akin to what is naturally there in the ecosystem, that's going to be really different than introducing 1 billion of one kind of organism. So I think the more we think like ecologists, the more we think about the dynamics of the ecosystem that is the gut or the mouth or the vagina or the skin or wherever there are lots of microbes, the more we're going to be able to make sense out of, the puffery that is associated with the promotion of certain treatments, and the more we're going to move into a system where it is useful.
No, definitely. And the conversation brings to mind a past conversation I had with a really, really fascinating guy named Steve Ranella, who he's a hunter. He's the one who introduced me. Have never been a hunter, but was introduced to it for researching my last book when I wanted to hunt and forage for all my food for a period of time. And he was talking about the sort of politicized response to reintroduction of, say, wolves, and how when you try to oversimplify it, just like carpet bombing your system with 20,000 pills of fill in the blank single type of probiotic or bacterium, that you could introduce, say, all right, there's wolves are dying at an alarming rate. They're nearly extinct in this one place. Let's add 1,000 wolves. Well, that doesn't really work very well because it throws the entire system out of whack, and then you have, basically a mass genocide of, their primary prey. And what he pointed out also, and I think this is kind of interesting, is that the microbiome people associate, I think, broadly with the gut, right? They think microbiome, they think I swallow pills, put them in my gut, and they're reading about fecal transplants and whatnot. But it's a lot, and as you mentioned, Jonathan, even in the GI tract, you have these vastly different populations in different areas. And what Steve pointed out, he said, people are against hunting wolves, for instance, because they're thought of as very sparse. But the fact of the matter is they're very, very, very overpopulated in very specific areas, and therefore the averages are misleading. So you should actually hunt and cull in certain places. But getting a little down the rabbit hole with this with this story. But I'd be very interested to hear, Jonathan, you comment on the just from the evolutionary standpoint, how do people go about figuring out what to aim for when it comes to that ecology, right? Because it's in a natural environment, you could say, wow, everything's going to hell in a handbasket. Something is wrong. It's kind of perhaps more obvious to pinpoint problems, but should we be looking to tribes in Tanzania or Papua New Guinea or the untouched parts of the Amazon, if anything exists like that, to try to, where the children haven't had antibiotics? I mean, how do you determine the menagerie of bacteria and the balance to target or to aim for?
Yeah, I mean, it's actually a very complicated question and a complicated sort of research topic. Certainly, understanding where our microbiomes used to be in the past is a very helpful component of answering your question. So if we can get access to, mummies and bog people and Iceman and other ancient samples, microbial studies of those samples are being done, and they're very important for placing what we see now in a evolutionary context. And other people are going out and looking at populations of just diverse sampling of the human population, but looking at populations that have been less exposed to, certain types of food or certain types of drugs or certain types of environments, and that is also providing that sort of context. I mean, we, in my research, in my lab, we actually work on the methods by which you would compare to evolutionary relatives or to ancient samples to try and figure out what the microbial community used to be like. And the reason you want to know that is twofold. You want to know, what the immune system and what the organs and what the blood and what the rest of the body sort of evolved to see. And you also want to know how it's changed with humans. Obviously, we want to know how it's changed in response to antibiotics and in response to changes in diet and in response to globalization and other issues. Now, that still doesn't tell us what the goal should be, right? Because we live in a world now that's different than the world we lived in 20,000 years ago or 20 million years ago. So, but it gives us sort of that framework to help interpret when you then do a study, say, of comparing people in Japan who live in Japan now to people from Japan who moved to the United States. There have been a few microbiome studies like that. And it helps you figure out what the disturbance is and what that might mean for our biology. But I don't think it's obvious how to say we should have a goal of making our microbiome like it used to be 20,000 years ago. I mean, we have different diets, we live different lifespans, we have different interactions with other communities, it's pretty hard.
Yeah. No, it seems extremely hard. Jessica, what are some common disruptors of the microbiome? So if it's certainly difficult to decide what the ideal should be, but what are we aware of that can disrupt or cause problems with the microbiome? So I've read certain, I wouldn't call them studies at this point, but that things like Splenda, artificial sweeteners, can cause issues with the microbiome. What are other common disruptors of the microbiome?
That's such a good question. So, I just sort of want to start off with that point about there is no ideal microbiome. I think that's important. There sort of aren't, people often ask also about, are there good bacteria or bad bacteria? And really, from an ecological perspective, you can't say, well, a good bacteria in the wrong place is a bad bacteria,
right,
or a good bacteria at the wrong time. So I think some of that exoticism about, let's go back to our ancestors is not, a way to find the right, to learn how to cultivate your microbiome, I guess. But in terms of disruptors, so there's, obviously, let's start off with the number one elephant in the room disruptor, which are antibiotics, right? We see this in the results of people who have taken our test. When you take antibiotics, a day later, your microbiome, there are whole, genuses of the microbiome, they're just not there anymore. Mm-hmm. And presumably they come back, as you test your microbiome, as your microbiome recovers from the shock of the antibiotics. But it's definitely, very clear that taking antibiotics just kills whole swaths of the microbiome. The study you're referring to about the artificial sweeteners was showing that it can be that artificial sweeteners can affect the microbiome in the way that sugar can. So I don't know if it's so much of a disruptor, but I think the big thing is sort of changes to the microbiome in terms of diet. So there's some really interesting research about people, what happens when people have celiac or people who are gluten intolerant eat gluten, and how that affects their microbiome, and how it just increases inflammation tremendously even after they've stopped eating gluten, presumably through the mechanism of microbes that are still there from when they ate gluten and are still causing inflammation. There's some interesting research around dairy that shows some similar things. So it's kind of all the usual suspects in terms of what we think of as disturbing our gut is also shown to have, impact, to mediate some of that impact through the microbiome.
Can I add just, there's another component that I view of as a really big, quote, disturbance, and it's more of a disturbance relative to what our bodies evolved to sort of expect, and that's, early in development, cesarean sections, for example,
right,
or feeding formula instead of breast milk, or excessive cleanliness as a child. There's a lot of epidemiological data and some microbiome data that shows that all of those things that, in essence, change the colonization pathways for how a young human being will get colonized can lead to longer-term problems. And I view that as a disturbance in the microbiome because, vaginal birth can be viewed as a delivery mechanism for microbes and as a component of it. Obviously, it's not the only thing, but breastfeeding is clearly intimately tied with development of the microbial community, and even playing in dirt and experiencing the microbial world in some normal setting. That's what we evolved in that type of environment. And whenever we disturb that, whenever we deliver by C-section or have antibiotics or don't breastfeed or are excessively clean or have all of these things that are not the normal developmental path, it's not that those are always bad, but they change the way our system sees microbes. And some of the time, our system's response to that is inflammation or problems with the immune system development or, other types of developmental abnormalities. And I think that...
Oh, sorry. Go ahead, Jonathan.
I was just going to say, I think that those should all be viewed as disturbances too.
Definitely.
So there's some interesting research about autoimmune disorders, and the hypothesis is that autoimmune disorders develop because the body doesn't have, sort of the hygiene hypothesis sort of on steroids, is that even autoimmune disorders are not caused by current lack of microbes could be caused by lack of microbes at a specific point in human development. So, you take antibiotics in your first year, it triggers something in certain individuals who have a genetic predisposition to develop Crohn's disease, for example. Now that part isn't proven, but I think it's very interesting because you want to look at microbial development on a timeline, not just what happens to be present in your gut right now, but what was present when certain events occurred.
No, that's a really fascinating way to look at it. And Jessica, you introduced me to a gastroenterologist that maybe we can name another time, but since I'll be talking about our conversation, I won't name her right now. Very, very bright. And she asked me quite a few questions, and one of them was about my antibiotic use when I was a child as opposed to adult, and how that could contribute or not to immune function now, even though perhaps I had, as I did, for instance, chronic sinus infections when I was a kid. So it's a really fascinating looking at the chronology of your microbiome to sort of follow the gingerbread trail to your current state, not just looking at the snapshot of the current fingerprint of your microbiome is an interesting.
So what I'd love to do is, I think, and maybe it's natural that I would think this, but I think that it would make a ton of sense to take sort of a fecal bank when, at various times in a person's life, so that when later we have the technology to create, artificially that community of microbes, you can give it back to you. So
that's a good idea.
Wouldn't that be awesome, right? You take your gut right now, when it's relatively good, if it is, or your gut, throughout certain points in childhood, you bank that sample. And then when we know how to recreate microbial communities better than we do now, you just say, oh, I want the gut of myself at 25, or, I want the gut, before I had an onset of Crohn's disease. Well, okay, great. We'll tell you exactly what was in it, and we'll give it back to you.
Oh, here's a question, Jonathan. Is that technologically feasible? I mean, if you had access to the nitrogen and apparatus to freeze it, like a sperm sample or stem cells or whatever, could you bank fecal matter, or would it just obliterate the?
No. So first of all, people are doing exactly that. They are, for their children, freezing fecal samples with the hope that either they will be characterized in some way, like with DNA analysis in the future, or that you could recover the living organisms from those samples. You can certainly recover living organisms from samples if they're frozen in the correct way. There's, this stool bank fecal transplant organization that was started by people from MIT that is doing exactly that, trying to store fecal samples for fecal transplants for future. There's definitely the technology to do this, at least at some level. I don't think we know how to identify which of the samples are the ideal sample, and not everything survives that freezing. So you can't necessarily recover the entire microbial community, but I know multiple researchers who are doing exactly this, collecting samples weekly, monthly, or something to that effect, storing them, characterizing them in some detail, and then anticipating as part of a research study, restoring some from the past if something goes wrong.
So this is something we offer to people. If you sample your microbiome now, even if there's nothing particularly interesting you find in your sample, although that's unlikely, it's possible, then you'll still have it. So we'll know what was in it. And maybe, I think where the technology isn't there yet is in, like Jonathan said, in bringing the microbes back to life. The Lazarus microbe doesn't exist yet, but certainly the characterization is there. So certainly you can say, here's what it was like. And when the technology gets to the point where you can actually then recreate that community or infuse that community in the proper way, that you'll at least know what was going on at that time.
Well, it seems like, as Jonathan mentioned, I mean, as we are able to identify and classify more microbes, and of course we're limited in terms of the big picture, the complete picture, by what we're able to identify, but and Jonathan, maybe you could talk to this, but it doesn't seem like, from a sort of usage of recombinant DNA and replication of these bacterial strains, like you could figure out the percentages and have sort of the, like synthetic poop made to order kind of thing, to implant at a later date and time. I don't know. That's pretty exciting. I know, I interviewed a woman named, Ph.D. Rhonda Patrick on the podcast not too long ago, and we were talking about banking stem cells and how, parents can keep the child teeth, the teeth that are lost by their children for stem cell banking. And it seems to make a lot of sense. I mean, I can imagine after this podcast, there's going to be some, intelligent and, proactive, tech millionaire who decides to have like a poop cellar right next to his wine cellar with just like a gazillion samples. Cause, unlike freezing eggs, right? I mean, humans, at least most of them seem to poop quite a lot. So there's no shortage of material. So
I just want to say that's something we promote to people, that you can have that sample. I mean, the cool thing that's interesting also about, banking poop is that, let's say the analysis methods of today are not the right analysis methods, right? Like we do 16S sequencing. You can also do full metagenomic sequencing, which is sort of finding everything that's in the sample. Let's say none of those work and we actually need some totally other method. If you at least have the sample, you can go back using the method of, 5 years from now, which is way better, and use that to do whatever kind of analysis that you need.
Yeah, no, definitely. I mean, I've been, since it's, just us on the phone here, and
a million other people, literally.
People. Hi. I have been banking, sperm on and off for the last 5 or 6 years. And, one could argue, well, like you're 36, 37, like it's already too late, pal. Like your sperm is definitely past its prime. And, be that as it may, which I suspect it is, we do not know what technologies or techniques may become available in the future for sort of rejuvenating or modifying those samples also, right? So there's no, from my perspective, it's just like, well, look, if you're spending hundreds of dollars a year on car insurance that you never really use because you don't get in an accident, what's the harm in spending an equivalent amount on storing biological material that could prove, even in a 10% chance, to be very, very helpful and could even save your life at some point or something like that.
That's a really good example on an individual level and also on a scientific level. I was talking with someone at in the UK about NHS banked samples, and how, because it's the National Health Service, they bank all their hospital samples. So they have fecal samples and blood samples going back to World War II.
Wow.
Like, how interesting is that, right? Like, from a personal perspective, yes, of course, you want to bank your own sample, but also think about being able to like license the use of those samples, or just give that data to a researcher that's studying something really important that your family could benefit from, or a future sufferer, or, I mean, there's just sort of possibilities are endless there to be able to take that data and make it valuable once you have it.
So let me ask sort of a curveball question. It's not too crazy. It's just unrelated to what we were just talking about. And the question is going to be, what do your close friends think you are world class at? And Jonathan, I'd love to maybe have you take a stab at that, if you wouldn't mind.
Social media. As far as I know, that's all they know about what I do.
And branding, phylogenomics.
Yeah, exactly.
Making up words that isn't on the list somewhere.
and studying weird microbes.
So within the world of studying weird microbes, what are you known for, would you say? I mean, what if in response to what would someone say, Jonathan's the guy you need to call to talk about that?
Yeah, I mean, I think it's using an evolutionary perspective in trying to study or design ways to study individual microbes or communities of microbes. So, a lot of researchers do great work on characterizing a particular microbe or a particular system, but they're focused on just that system with a lot of their work. And what I really am known for, I think, and what I specialize in is saying, yeah, that's really interesting. But on top of that, it can be really helpful to understand the history behind that organism. That can tell you what direction it's going, where it came from, and allow you to make predictions about where things are going in the future. So I think my whole career has been basically taking that one theme, adding an evolutionary perspective and applying it in a lot of different areas, and just hopping between, genomics, so that's phylogenomics, and microbial communities, and functions of microbes, and how organisms survive in extreme environments, and what lives in and on humans or on other organisms, and always saying, oh yeah, but what about the history?
Right. I really want to ask you a question, but before I do, because I suspect it might fit in this category, what are the questions that you get asked from an evolutionary standpoint or evolutionary biological standpoint that are most irritating to you?
What is the most ancient microbe alive? When all microbes are all equally ancient right now, none of them are old. They all grow and replicate. That always drives us. That always drives us crazy. Another is why the classical intelligent design questions. I guess I don't find those as irritating. I get a lot of them, but I don't find them that irritating because they're so common.
Right.
And I mean, I think another really thing that I would say completely drives me crazy, and I get this question a lot, is, why study that in microbes? Microbes are so simple.
That's a rather condescending way to put it.
Yeah, I know, and just it has so many layers of, inappropriateness and condescension. I don't even know how to respond sometimes.
You just sidekick them. That's usually, I find that pretty appropriate. So the question I was going to ask you is, how did Adam and Eve ride to their wedding on a dinosaur? That's really no, that's not my question. I'm kidding. The
microbes made them do it.
The microbes made them do it. That's driving behavior. They, those dinosaurs had a lot of sugar. The question I wanted to ask you, which is off base, but I just get asked this so often, and I would love to have a more informed answer to it, is, and if this is outside of your expertise, please, you feel free to dodge. But how should one answer the question, or let me rephrase it. What do you think of the paleo diet or vegetarianism? Let me just throw those two out there.
The vegetarianism, and I hate all rules, first of all, as in all sorts of different environments. So anytime someone says I'm going to apply some rule to my life or to science or to something else, I kind of get irritated by it. But the paleo diet, I think, is a great example of something that is really interesting from a conversational point of view, to think about how people used to live and what they used to eat, but to apply it to our modern lifestyle as though it's going to somehow be magically perfect seems a stretch to me. Let's put it that way.
Are you trying to tell me that cavemen did not eat coconut macaroons?
No, I don't.
I'll be very disappointed. No, I'm kidding.
I think cavemen did a lot more than we appreciate. As they just discovered those supposed etchings on a shell from 500,000 years ago, and we're always finding new, interesting behavioral patterns in Neanderthals and in cave people. I think we underappreciate what the ancient human lineages did, but I certainly don't think imitating everything they did is the right way to go.
Yeah, and I don't have a particular dog in this fight.
Yeah.
What about vegetarianism?
Yeah, I think from a health benefit point of view, I think it's not necessarily. It's also sort of it could be good for some people, not necessarily good for every person. I think that's a separate issue from the political point of view. I think
of course.
very clearly the current mode of production of meat on this planet has enormous ecological and health problems associated with it. That doesn't mean like going out and raising your own cattle and hunting and other things wouldn't, be relatively not as damaging to the planet. But, production cattle and chicken farms and other things are causing enormous problems with global climate change and with antibiotic resistance and with the origin and evolution of pathogens. I mean, they're just a nightmare, basically. So vegetarianism, if you're doing it, from a political point of view, I can understand it. I'm not one, but I can understand it.
Yeah. No, I think just to underscore something for folks, if you avoid antibiotics but don't discriminate your sort of animal protein consumption, you probably are taking antibiotics.
You are, not
yeah.
Probably. You are.
Yeah. And so it's what's important to realize, I think, among many other things, it's kind of a haughty way to start a sentence, but I'll start it with that way anyway, is that people say you are what you eat, but you should take it a step further. And I think of it as you are what you ate, right? So you having factory farmed salmon that was given antibiotics may not be as good for you as eating locally grass-fed beef, for instance, nor is it necessarily better for the environment. So I think taking it, sort of looking at the secondary and tertiary steps is important, or preceding steps. You mentioned a distaste for rules, and I want to talk about this for a second, because how do you distinguish, and maybe you dislike both, this is a fine answer, but I find it very, for me to be maximally productive, I find applying constraints very helpful, using different types of constraints. So how do you personally distinguish between rules that stifle versus constraints that enable?
Yeah, I mean, it's funny, I just had a 2-hour conversation with someone about this yesterday. I think that the difference is whether or not you're going to apply them blindly, or whether or not you're going to apply them with an open mind and intelligence. And the way I view rules is they're applied blindly. And the way I view constraints, like you identified, is sort of like a slight difference in the probability that you would allow yourself to not follow them.
Right. So one is the "I told you so" because I told you so school of thought versus sort of strong, like a hypothesis weekly held, or guideline weekly held. Very interesting.
So just as an example, I'm a type 1 diabetic. Mm-hmm. Been on insulin for 30-something years. I tried many times to have rules about, don't eat dessert, or do this type of diet, or take insulin at this time. And I've found, certainly in my life, that I never can follow those rules anyway. And it just became completely stifling to worry about whether or not I was following those rules. Whereas, if I say, have dessert when you want it, but don't have it too often, and be careful about it, I view that as a constraint, and it works much better.
How did if you don't mind me asking, how old are you currently?
Gosh, that's a good question. Should I know this? I am 46.
So, do you have any hypothesis, or maybe how you did you have type 1 diabetes prior to being 16, and it was just undiagnosed, or did it have a sudden onset?
When I was almost 16, over a period of maybe 4 or 5 months, I slowly started to waste away to the point where I was had lost about 45 pounds, and was probably within a few hours of being dead when I finally went into the hospital. And the sad, funny, interesting part of all this is my dad worked at the NIH and was trained as an endocrinologist. And I had mentioned multiple times that, something's wrong, I'm really tired, I'm thirsty a lot. And, he was blind to he was a researcher, so he didn't practice much anymore. But, I knew something was going on, but I didn't quite know exactly what. And it's like the frog in the frying pan thing for me. It happened over such a long period of time that by the end, it was ridiculous. I mean, I was peeing every 5 minutes and drinking drinking, 40 liters of liquid a day to try and keep up.
Wow.
And completely ridiculous, right? And it ended when I was on a backpacking trip, my first one in my life, and I was putting my face into puddles of mud to drink the water. And, at that point, something finally clicked in my brain, like, this is wrong. And, insisted on getting checked later that day when I was, on the edge of diabetic ketoacidosis.
Wow.
And, yeah,
Wow.
it was pretty messed up.
What inspired you to become a scientist, if that's how you would sort of characterize yourself? I mean, obviously you have a tremendous amount of scientific and research background, but what inspired you to take that path?
So I'm from a family of dorks. In particular, my grandfather was a physicist, did X-ray crystallography and other types of sort of theoretical physics, and he just talked about science all the time when I was a kid. And if you go back to what Jessica was talking about earlier, I was a birder. I participated in some of those citizen science bird activities when I was a kid, doing Christmas bird counts and Thanksgiving bird counts, and I got really interested in natural history because of that. And I actually went to college and I was an East Asian Studies major for a while, but I realized that I was too interested in biology. I actually took a course with Stephen Jay Gould on evolution and realized, I can't do East Asian Studies. I just love science. I love biology and I love natural history. And that's what I view I'm still doing. Natural history of microbes instead of birds, but same general idea.
And that was at Harvard?
Yeah, I was an undergrad at Harvard, and I was taking Japanese classes and I was taking, East Asian history courses. And then as a non-science major, you had to take some science courses. And one of the ones they offered was this course by Stephen Jay Gould. And I had read a lot of his books, so I thought, hey, that's kind of cool. And like within half an hour of one of his lectures, I knew that's what I wanted to do.
Wow, talk about that's quite an inflection point.
Yeah.
Well, you and I definitely perhaps another time have a lot to talk about. I was an East Asian Studies major undergrad myself, focus on Japanese, and planned on transferring to the neuroscience department. Specifically for a number of professors, Barry Jacobs, chief among them, and could not. And I think that animal testing is hugely important, but I could not bring myself to pay the dues in the lab, which involved, I guess they call it perfusing, 30 to bleeding to death, i.e., 30 to like 40 rats a day. And I was just like, oh God, I can't do it.
Thus, why I work on microbes.
Yeah, right. Right. They don't make a lot of noise when you euthanize them. What about Jessica? What about yourself? How did you come to be to found uBiome? I mean, what sparked that interest? How did
so
that?
I came to this a lot later. I think I was always very interested in science. My dad's a chemist, and we used to talk, physics and science and chemistry. When I was a kid. So I was very interested in science as a child, but I didn't my early science courses was a lot of sort of recreation of early experiments rather than doing your own experiments. And my passion was always for learning new things based on science. So I went a totally different route. I studied economics and computer science at Stanford. And then I got a fellowship to go to Oxford to study, at the Oxford Internet Institute, and then, in other departments at Oxford. And I basically sort of took computer science and economics and put them together to be computational social science. So looking at the mathematics of social networks, kind of taking a different approach to quantifying human behavior. So, in economics, there are very specific mathematical approaches that are often used. And I was really frustrated with those. So I thought, okay, let's do something different that's still quantitative and what is now called, big data or data science, but focusing on using different computational methods to understand social science data. And so I was doing a PhD in that, and I was part of a program called Startup Chile, which was a program to go down to Chile, the country, and they give you $40,000 and a visa to Chile so you can become an entrepreneur and start your company in Chile. So I went down there to work on commercializing some of the ideas that I'd had about social networks when I was as part of my PhD. So how you can measure social networks better, how you can determine which ones are more valuable than other ones. And it was kind of interesting. But then I met my co-founder, who was doing his PhD at UCSF in biophysics. And
This is Zach.
this is Zach. Yeah. So Zach was doing his PhD in biophysics at UCSF, working with microbes and doing some very similar types of mathematical techniques to what I was using in social science. And just sort of a plug for social science. This is a rare area where the methods are better in social science in a lot of ways, just because there are many more data points. So if you're looking at data from LinkedIn or Facebook or something, you have millions of data points, which is, often hard to get in a biological setting. So we just started talking about this, and I just thought, I could take these same skills and apply it to something like genomic data, where you could just have this tremendous impact on human life. And it's led for me into this passion for science and public health that I was always kind of nascent, but I just didn't, I don't know, the academic structures just didn't help me to apply it in a way that felt meaningful to me. And just the idea that you could take the same data science techniques that you use to understand, LinkedIn data, which are interesting there, and there's a lot of value to be gained, but the microbiome is this whole other area of human endeavor. There's, it's a whole new organ in the human body that had never been discovered before. And being able to take those same techniques and apply them to something that's sure to yield some impact on humanity was just what I had to do.
How, without, of course, disclosing the secret sauce and anything you do not want to have public, where do you hope uBiome to be in 3 years? What's if you could comment on that, or you can no comment it. That's fine too.
I mean, I think we've been pretty open about what our mission is and what we want to do. And we want to involve the public in science by doing large scale studies that involve the public, and then to take the products of that research and turn them into useful things that people can use. We will probably not be the people turning them into those things. So through partnerships with pharmaceutical companies, consumer goods companies, other companies that can actually turn this knowledge into something valuable. This isn't selling people's data. This is sort of taking the insights we learn from the people who are giving us data and then taking those insights and turning them into something meaningful. So that's kind of our focus. So it's something like, this is kind of a terrible example, but I can't think of a better one. It's sort of like Shazam. Use Shazam to figure out what a song is, and then they take that data and they don't sell what songs you like to anyone. They sell what songs are trending and what songs should you be writing. It's something similar to that. We sort of will figure out what is going on for different populations over time, through the cooperation with the public, and then we'll be able to sort of take those insights and say, okay, this is clearly useful in this way, and helps, and give the information to someone who can best make use of it.
Very cool. Yeah. Well, the reason I backed uBiome is specifically because I'm waiting for you guys to develop The Rock in a Pill, the professional wrestler / actor. So I want to have his microbiome.
We'll do it for you.
Since he's like 347 pounds of pure muscle, I assume that will be replicable.
Oh, it's coming soon. Soon.
Yeah. All right.
I mean, in all seriousness. I mean, I don't know if you get The Rock in the Pill, but I think there's just a ton of really interesting and useful things that can be made from our understanding of the microbiome more quickly than in other realms. If you want to take a whole other approach to drug development for cancer, for example, there's just a tremendous amount of research and experimentation that needs to happen. But we're sort of these natural laboratories for microbes, in terms of us all having different ones that are doing different things, and we can learn from that much more quickly. That's at least our hypothesis. Than you can sort of take other approaches to developing interesting products from the microbiome.
No, definitely. So I would love to ask a couple of odd questions of you guys. These are a little bit of a shift in gears, but these are what I usually just call the rapid fire questions. But that doesn't mean that the answers have to be rapid fire, but we can try. The first question, this usually isn't the first one that I ask, but Jonathan, I just love I love the fire in your belly, so I'm going to ask this one. When you think of the word punchable, who's whose face is the first one that comes to mind?
Jim Watson.
Jim Watson. Oh my.
Whoa,
No,
that one.
No, this is amazing. All right, so please elaborate.
Well, I don't know if you saw the whole thing about him selling off his Nobel Prize medal recently because he's so impoverished now because the whole world hates him after his racist and sexist commentaries that he made a few years ago associated with the talk, and now he just has to sell that Nobel Prize to raise some funds to buy a painting.
To buy a painting?
Yeah, to buy a painting. That was one of the things he needed to sell the Nobel Prize for. And Watson did some interesting stuff for a while, but in all honesty, he's kind of a deplorable character these days, and I don't think there's anybody that comes to mind quicker than him in terms of punchable.
Wow.
That's an amazing. No, that's a fantastic answer. The painting must be have high nutritional value.
Yeah.
Must be a hell of a painting.
Yeah, or like very thick for shelter.
Well, even more amazing, some Russian oligarch bought the medal and is giving it back to him.
Wow, that's much more altruistic than I would usually assume Russian oligarchs to be. Go figure. Actually, so Jessica, I'll come to you in a second. But Jonathan, what is the book or the books that you have gifted most often to other people?
Oh, I know the exact answer to this. I give it to tons of people. It's a field guide to the birds of North America by National Geographic.
Cool.
And it's like the only book I give to people.
Well, these rapid fire questions are really working out here. Jessica, what about you for books?
Oh, that's a good question. So I guess it's kind of boring, but I give Hemingway's short stories to a lot of people. I'm a big Hemingway fan. And,
why are you a big Hemingway fan?
Oh, he's just such an artisan of the story. Mm-hmm. Like his stories are so well crafted, and it's one of those things where you look at a work of art and you can't exactly figure out how they did it, but it's perfect.
Yeah.
So I admire the craftsmanship so much.
Yeah, there's not a lot of bloat in Hemingway's work.
No, that is true.
Yeah, if you're a Hemingway virgin, The Old Man and the Sea is a pretty good place to start, I think. Well, the short story is also fantastic. Yeah. Jonathan, do you have any particular morning rituals? What does the first hour of your day or 2 hours of your day look like? Is that is it pretty standardized?
It's very standardized. My wife probably gets up first, and we have about an hour before our kids get up, and usually we make coffee on a stovetop coffee maker, and we drink coffee, and we sometimes sit there and space out, and sometimes we talk about the plan for the day. And usually our cat comes over and hangs out with us, and that's what we do. That's what we do for an hour.
When do you wake up?
Her alarm goes off at like 5:55 in the morning. I'm sometimes up before that, sometimes up after that, and we have about an hour before the kids get up.
Now, okay. Now this is amazing. I may have a soul connection with your wife, which sounds weird to say since we don't know each other that well. Why 5:55?
I don't know. She really likes to have time to veg out and to think and to process before dealing with the ritual of getting the kids fed and off to school. And I don't think an hour was enough. So an hour and 5 minutes is, like the bonus time.
Wow. So 5:55, I don't know if I've talked about this publicly. I won't digress too far, but, so 5:55 PM was the time that I finished editing, a book I wrote called The 4 Hour Body, which was a monster. I mean, it was almost 600 pages after cutting 250, and it was the exact time that I finished my last line of editing in the book and was ready to mail off the final thing. So I always take a screenshot on my iPhone whenever it's 5:55 because it's sort of my good luck omen. Anyway, that's amazing.
That's awesome.
Give a high five to your wife.
I will take screenshots now when I get woken up by the alarm.
It's going to be a lot of screenshots.
Yeah. I'm sure I won't be able to think well enough to take more than a couple.
Not photos, screenshots.
Do you now, coming from a family of scientists, how do you think about parenting differently from your non-scientist friends, if at all?
Yeah, I mean, I'm not sure at all. You know, we went through, when our first kid was born, our daughter, we went through the, you know, like read a bunch of books, like they're going to tell you exactly what you're supposed to do related to parenting. And most of it is just, you know, something that worked for one person and, you know, it didn't necessarily work for us. And again, it goes back to that rule thing. If you follow those books as a rule, they're horrible. If you follow them as a constraint and guideline, they're pretty good. So I think my wife's also a scientist. She hasn't been working in the lab recently. We say she's a child developmental biologist. But so we're both scientists, and we have a lot of scientists in our background. But we try not to either force science onto our kids or obsess about scientific approaches to everything. We're much more sort of holistic about how we deal with the kids, in holistic in the sense that we try to, you know, talk to each other about things and think of, you know, common sense approaches as opposed to digging into every possible scientific study that could have been done.
Right. And it also brings up a good point. Just the books on parenting comment is this is a good example of sort of where media, I think, can get things totally ass backwards, where they might say, well, we looked at the data and you had 100 people who read parenting books and 100 people who didn't, and the people who read parenting books turned out to be better parents. Therefore, parenting books make you better parents. And it's like, well, it could just be that the people who are proactive enough to actually go buy books on parenting are going to end up being the better parents anyway, because they care more and so on and so forth. So, that seems to be the feedback I get from a lot of my friends who are parents. I am not yet, but I'm certainly looking for the cliff notes to the extent that I can. Well, one more question for you, Jonathan, and then I'm going to start raining questions on Jessica for a bit. But is there a particular defining moment of your childhood that you can think of, or was there a defining moment in your childhood that helped mold you into who you are?
I mean, I think, you know, we've already talked about one, which was the, you know, being moments away from probably going into a coma and dying from diabetes, which really had a massive impact on lots of things. But I think unquestionably, I can tell you one very specific moment that had a huge impact on me as a child. When I was almost 10 years old, or just 10 years old, we went to Kenya to visit my uncle, who was studying baboons there for his Ph.D. in anthropology. He was doing field work in Amboseli. And we went camping in the middle of the Masai Mara and other parts of Kenya, you know, with lions wandering around outside. And one day we were driving through the savannah, and my uncle said, "That's weird," and pointed way off in the distance to this gazelle that was doing something strange. And we stopped the car and waited. And slowly, over a period of maybe 15 minutes, a cheetah came from over where that gazelle was and walked about 5 feet from our car. And then we watched it do the cheetah sprint. It didn't catch the gazelle, but it did the full, 60-mile-per-hour sprint. And that moment was so awe-inspiring and transformative to me, to just think more about the natural world. I mean, I was kind of into birds then, but I wasn't really thinking about it. But that, just seeing the dynamic system in the savannah and seeing this incredibly beautiful, graceful predator coming out of nowhere, that I still remember that moment.
Wow, that sounds incredible.
That's an amazing story.
What time of day was that?
It was in the afternoon.
Okay.
I mean, I don't know exactly when, but
I was just painting the picture in my mind, because it's just like, wow.
It was incredible. And by the way, as an aside, we're now looking into doing studies of the cheetah microbiome, because I have to return to my roots.
I was hoping you would say that you have adopted a cheetah and put a saddle on it, like Harold and Kumar go to White Castle.
Yeah, that too, but I can't tell people about that.
Right, yeah. Legal only in several states, not all. Jessica, when you think of the word successful, who is the first person who comes to mind for you?
Oh, wow. That's a really good question. There's so many different ways to be successful. I think, so no particular name comes to mind, but what I really admire is when people have come a long distance from where they used to be to where they are. Mm-hmm. And I think sometimes the people who've done that haven't come very far on a global standard, but they've come very far from where they used to be. So I always love, reading stories about people who, were in prison and then totally changed their lives, or people who, came from really modest circumstances and really did amazing things with it. So I think for me, like thinking about that distance that people travel is what makes them successful.
Okay. No, that's a good answer. I think that, I guess it's what Hurricane Carter. I mean, there are a lot of course, many people who are inspiring in that sense. Who do you aspire to be like or to emulate as a founder or CEO?
Oh, that's a great question. Oh, wow. So I'm not, this confession, I suppose, I'm not a big worshiper of the sort of canonical Silicon Valley CEOs, Steve Jobs and Larry Ellison and, even Sergey Brin and Larry Page. I think a lot of these things are sort of, Six Sigma events that happened in a particular place and particular time, and sort of slavishly saying, well, Steve Jobs did it, so it's a great idea for us, is kind of not the best way to go.
Right.
I think the people that I try to emulate are people that are one step past where I am, not people that are 10 steps past where I am. So they're probably people you wouldn't necessarily know, but they're like, they've kind of gone to the next stage where I aspire to be, and then the next stage after that.
No, that's fine. Any particular names? Would love to hear.
No
some
names
specifics.
are sort of coming to mind.
Okay. No,
I do a lot of reading and talking to people, but no names are coming to mind. At least no one who I would think it'd be okay for me to name them on a podcast.
Yeah, no, that's all right. How do you find those people?
That's a good. So I find some of them through our investors. We find people that have sort of been funded at the next stage or who have done the things that we're trying to do. I find them at conferences. I also just sort of find them through researching online and trying to think, who has done the thing that I'm just about to try to do, and or maybe the thing that comes right after that, and try to talk to them. I think you were talking about superpowers earlier, and I think if I have any sort of superpower, it's sort of that, figuring out who is doing the next thing and being able to talk with them and learn what they know. That's
a good superpower. What are the questions you like to ask such people?
Oh, so I love to ask how things were done. I think sort of a lot of these things, especially when they're reported in the media, are like, so-and-so is an amazing CEO and they did this crazy thing. But when you actually find out how it happened, it's because their college roommate was doing it also, or their professor told them how to do it, or there's, some trick that they know that nobody else knows. Mm-hmm. So I like to ask sort of questions around what that trick is. Like how exactly did this happen? Where did you first find out about it? What happened specifically? And I think that's sort of how questions are really important. I also like to ask values questions because I think that leads to a lot of interesting answers about what was fundamental to what happened as opposed to what was merely incidental.
What would be an example of that?
Oh, so there's a CEO that I sometimes talk to who has a company that has a really strong culture, and I ask a lot about what was really fundamental in drive, like what things did they not do that they could have done that perhaps they even wanted to do that they didn't do in order to have the culture that they have. So what thing cost them? What was the sort of, where was there some sort of
what sacrifices did they
make? Exactly. Well, what sacrifices they make sounds a little bit cliche. It's like, oh, well, I didn't go to the beach, or, I didn't spend time with my cat or whatever, And I'm thinking about it more in terms of sacrifice.
It was a dog culture. You got to choose sides.
Right, exactly. I'm thinking more about things like what decision did it hurt you to make, but you're glad you made it?
Can you give an example of any of the answers?
Oh, yeah. So I think one particular answer is sort of trusting the people around you in ways you might not necessarily think of to do.
Like what?
Oh, well, in terms of just having a company culture that's very open and very trusting of the people that you work with. For example, I mean, this is something that we try to do at uBiome. Like we have, a lead data scientist we hired. He's a theoretical physicist. He's, just got out of his postdoc. And he's never managed a team. He's never written code. He's never done any of these things. But we have him doing all kinds of stuff. He's hiring people. He's doing all kinds of things just because he wants to and he can. So I think there's sort of a profound respect for that I have and that I learned through these conversations based on on letting people do the things that they want to do if they seem like they can do them.
Mm-hmm. We can dig into that maybe in a round two. So that's a big subject. Yeah. Jonathan, do you have any favorite documentaries or movies that come to mind?
If you want nonfiction like documentaries, in terms of movies, I've been watching over and over Shackleton movie. It was like a made-for-TV or cable movie about Shackleton. It's incredible. It's just the best thing out there that is about the Shackleton story that currently exists right now.
What do you like about it? This particular coverage or storytelling?
Well, the story itself is remarkable in so many ways. I'm really interested in exploration. So, the great stories of all the explorers over time I read a lot of, and I think the Shackleton story obviously is pretty incredible because, they were setting out to do one particular thing. It didn't work out the way they wanted it. And despite getting stranded on the ice and, having a very high probability of death, they all survived, right? That's the amazing thing about the Shackleton story is they all survived. They managed to navigate across horrible waters with very few tools and traverse, incredibly difficult circumstances. And over 2 years, even though they sort of fought with each other, they did mostly stick together. And it's just this amazing sort of story of human spirit and persistence and exploration. And I read a lot of these stories about, Antarctica and Arctic exploration. And prior to that, exploring the world around us, and the Shackleton one just is pretty incredible.
So this is weird because I have a sore spot for those kinds of exploration stories. I haven't watched the Shackleton documentary, but I love sort of the, Beryl Markham and Amelia Earhart and that whole sort of, early flight stories. I'm kind of a geek about those. I want to throw out a plug for a movie. I don't know if you've seen this, Tim. Have you seen the movie The Edge with Anthony Hopkins?
No, I have not.
Okay, so this is a movie nobody
wait a
has
second.
seen.
Is this about the Indian motorcycle
No.
tests? No, okay.
So this is about so Anthony Hopkins is like this sort of this perfect character. He's a billionaire scientist who's exploring the Arctic, right? So it's kind of this like sort of out there setup where he's, this billionaire scientist who doesn't spend enough time in the real world and he gets lost in the Arctic. And there's all sorts of interesting subplots, but basically he needs to find his way back to civilization and kill this bear that's trying to kill him.
Oh, and I saw a trailer for this. Okay, so
this was never this movie is not famous. I don't know why. It has Anthony Hopkins and Alec Baldwin and Elle Macpherson in it, so you'd think it would be something everyone has seen, but no one has seen this movie, and it is my absolute favorite movie. It is just sort of this great story where not only does he sort of survive by his wits and defeat nature in a way, he also, his companions are not on his side. Not to spoil it too much.
Yeah, no, don't spoil it. I'll watch it tonight.
And they're not on his side, and he manages to save them too. So there's this sort of this altruism to it as well.
I'll watch
it's
that.
Really
I'll
good.
probably watch that this evening. And Jonathan, I've not seen the documentary on Shackleton, but the one of my favorite things, maybe my favorite thing, just not knowing much about that story, one of my favorite things about it is the classified ad, and I'm looking at a copy of it, and the classified ad, this is what Shackleton used to recruit people to help in this journey, is headline, Men Wanted. And then the subhead is, for hazardous journey, small wages, bitter cold, long months of complete darkness, constant danger, safe return doubtful, honor and recognition in case of success. And then his name and address. Just so amazing. I feel like most job descriptions should have disclaimers like that, and the turnover would be a lot lower.
So
in
what
the
about
movie,
Sorry, I was just gonna say. So one of our investors, someone you know, Tim, loves that ad and said we should put that in all of our job ads.
Oh, I totally agree. Oh man, Jonathan, what were you?
I was just gonna say in the movie, Kenneth Branagh is Shackleton, and there's this great scene where someone is coming in with that advertisement to apply for the job. So it's just completely.
Wow. Okay.
Well, I heard that he was inundated with applications. I don't know if that's true or not.
They were drowning, and they literally got they had to hire people to go through the mail because they got so many applications.
That's so awesome. That's great. And I mean, the great thing about that disclosure is that they can always pull out the "well, I told you so." Like, you can't really complain about the darkness. I fucking told you in the classified. I mean, this wasn't a hidden risk. Jonathan, if you could change or improve one thing about yourself, what would it be?
Better organization. I'm not really good at planning things or keeping everything organized.
Do you think that the hatred of rules is related to that? Or?
Let's psychoanalyze Jonathan.
Should I lie down on the couch in my hotel room now?
No. I'm not qualified.
I think that there's a very strong correlation there. Whether it's causal or not, I don't know. But yeah, I mean, I think I do a really good job with getting things to happen most of the time, but I could definitely use much more effort on, a personal assistant or some type of planning.
So, okay, now this is, okay, so I don't let journalists follow me for what they want to be a typical day, really ever, because I find people expect me to be this sort of paragon of efficiency, and I run my life like Spock, and it's just this incredible sort of Swiss watch of productivity. And I'm like, if you came and watched me, you would just ask yourself all day, what the fuck is this guy doing? And but despite all of my flaws, I get a fair amount done, and it's for, reasons that I could speculate and talk about, and I have talked about ad nauseum, so I won't do it now. But you, Jonathan, you've done a huge amount, accomplished a huge amount. So what allows you to compensate for this lack of organization and get a lot done?
I think what allows me to sort of compensate for it most of the time is that maybe it is the lack of the rules. I have a big picture of what's happening in my lab and in the work I want to do and in the projects I want to do. And I'm not always obsessing with, am I following the right path for a professor, or am I applying for the right jobs, or am I submitting to the right journal? I do what I want to do. And it's worked out really, really well for me. And I think that in research, I know a lot of colleagues who spend a lot of time worrying about what they're supposed to do as opposed to what they want to do. And so I think because I don't spend a lot of time thinking about what I'm supposed to do, it works out a lot of the time that I get a lot done because I'm doing things I love and excited about, and I never worry about what I'm supposed to do. But, I confess, things slip through the cracks with that approach. And, most of the time it doesn't matter, but, every once in a while it does.
No, I like this. This reminds me of something that I read by one of my favorite writers, Neil Gaiman, who's a fiction writer, and he gave a commencement speech, and I think it was simply called "Make Good Art." But he talks about, I think he talks about in this particular commencement about sort of the big picture of moving closer to or further away from the mountain, which is this major goal he had of, I believe it was just being a full-time writer, and a lot slipped through the cracks, but because he had that one North Star, it sounds like, as you do with, say, the sort of overarching goals of the lab or otherwise, he's been able to create this incredible career, even though a lot has fallen through the cracks. So speaking of commencements, on a related note, and then I'll let us wrap up. This has been a lot of fun, and I want to be respectful of both your evenings. Jessica, if you were giving a few pieces of advice to your 20-year-old self, what would the advice be? And Jonathan, I'll ask you the same as well after this.
So I think my advice to my 20-year-old self would be about confidence. I mean, I think I've learned so much through working on my Ph.D., through starting this company. I've just sort of gained such a broader perspective. And I think I just didn't have confidence in myself and in what I could do and in sort of the level to which I could be pushed and still excel. And I think I would just tell my 20-year-old self to just not be so afraid of trying things. I think that's sort of number one piece of advice. And I think the second thing would be to really think very broadly about what's possible. That's kind of related, but just sort of not only think you can do it, but think about that there are many things possible in the world to do that I don't think I thought of when I was 20. And all the sort of standard, like buy this stock or
buy Apple, right?
Right, exactly, buy Apple.
What were you so? Why did you lack confidence? Why were you intimidated?
I've thought about this a lot. I mean, I think that some of this is sort of that traditional female sort of stuff where and I see this all the time. And when I see it in people, I always comment about it because I kind of make a point of it. Maybe I'm annoying about it. But I was just talking to someone today who was like, well, this is actually our director of operations. And she was like, well, I don't know if I can do that. Maybe it'll be, maybe I should just wait and then and ask them if it's okay. And I'm like, no. Women wait and ask. Men just go do it and then apologize for it later. And I think it's just like, it's true. There's sort of this like very female, like, oh, wait, and you'll be rewarded. Like that CEO of Microsoft was saying, women should just wait and not ask for raises. And then karma will give them money. And I just feel like he actually said that. And I think we're just socialized to believe that. So I think that's a big part of it. But I think another part of it was just I don't know. I think another part of it may have been sort of socioeconomic. I think there's sort of this like upper middle class, feeling of power that I learned at Stanford that I didn't know before that. And there's sort of this sense that you can get close to the seat of power. I remember when I first came to Stanford and I just, you meet people that you read about in the news. And I just didn't know anyone before who'd ever met anyone who'd been in the news. And you go to a talk and it's like the Secretary of Defense or something, or it's, it's Steven Pinker, or it's somebody that's just like, you've read their books, but the idea that these are actually real people who were not perfect and didn't do everything perfectly. I think as a child, I sort of imbibed this very my grandparents were immigrants after the Holocaust, and my parents sort of grew up in that kind of milieu in New York. And when I there's always this idea that if you're perfect, if you work hard and you're absolutely perfect, you too can succeed. And when I met people who were like the most successful people in the world, and they had lots of problems and were also very anxious about lots of things, I thought, okay, I could be one of those people.
Right. You're like, oh, okay.
I can do that.
I get it. These are human problems, not
exactly.
me
and
problems.
I just didn't know that growing up. So that was a big revelation for me.
And I want to just underscore one thing you said, and it's always a danger for me to say this because I look like American History X, so it's a sensitive subject. But I was having a conversation once with a very, very, very successful female executive, and the topic, this was a group conversation among friends, and the topic came up about salary discrepancies. And her thought, and I'm not saying this is certainly not the only perspective, but she was massively, she makes millions and millions of dollars a year. And she said, the reason women make less oftentimes is because they don't ask for more.
Yeah. Studies have shown that. That's not just her.
Yeah. And, the asking for forgiveness rather than asking for permission, I think, is a really important one. Jonathan, I'm sorry. Go ahead. Didn't mean to cut you off.
I was just going to say, this is very controversial because, Sheryl Sandberg's point about leaning in, but I think she's absolutely right. There are all sorts of structural inequalities and all those things are all true, but being able to just not stop yourself is at least a first order approach that you can take where other people may stop you, but at least don't stop yourself.
Right. No, absolutely. Jonathan, what advice would you give your 20-year-old self?
I would tell myself to bank all my fecal samples.
With uBiome, obviously.
And they'd be like, what? Like in an ice cube tray? Yeah, right. Like, how do I do that?
So weird. No, I mean, just like I don't plan things very well, I also am not very retrospective in thinking. But I think that the thing that I wish I had done more of, and that I think would be, really good in general, is to trust good people and ask them advice. I mean, I have asked people advice over the years, and when there are people that I really trust and people that seem to be good human beings, that advice has actually been really good a lot of the time. And I don't think I've done that enough. I think that, a lot of the time I try to work things out in my head, through some logical flowchart or, because I'm so damn smart, right? I think asking for help from the right people is a really good thing, and I wish I had done that more. And related to that, something I also wish I had done more, and I try to do much more now, is to thank people who help you. There's lots of people along the way in life who do something small or something big who have helped me and help everybody else in a lot of ways. And, in science, we stand on the shoulders of others, and, it doesn't hurt to thank those people.
That's great advice. Well, on that note, thank you both so much for taking this time. This was a really enjoyable conversation for me, and I hope that that's true for both of you as well. How can people learn more about what you are up to? And we'll go back to Jessica here. Jessica, where can people learn more about everything that you're up to?
Yeah, so as far as uBiome goes, you go to uBiome.com.
That's with a U?
With a U, yes, I should spell that. It's uBiome.com. So just go to uBiome.com. There's interesting information about what we're doing. Our blog is there. You can also purchase kits to better understand your own microbiome, and all sorts of interesting stuff you'll find there.
Just mailed one off myself today.
Thank you.
Back to you
we
guys.
look forward to receiving your poop.
And are you on Twitter? Any other social?
Oh yeah, so I'm
outlets?
on Twitter at Jessica Richman. So that's just my name, JESSICARICHMAN, like a wealthy man. And uBiome is also on Twitter at uBiome.
Very cool. And Jonathan?
Everything associated with phylogenomics. That's my Twitter handle. My personal blog is phylogenomics at Blogspot. My lab website is a phylogenomics WordPress site. So the best way to find out about me is probably to just Google phylogenomics, PHYLOGENOMICS, and then most of the stuff that comes up is something I've done.
Awesome. Well, guys, this was a blast. I really appreciate it. Have a wonderful weekend, and to be continued, hopefully.
Yeah, you too. Thanks so much for having me on.
All right.
Yeah, thank you very much.
Thanks, guys.
Okay, bye.
Bye-bye.
Okay, bye.